This lesson on applications for clinical trials in the context of chemistry manufacturing and controls focuses only on Canada and USA. We will describe the information to be submitted in support of the quality portion of a clinical trial application to Health Canada to ensure safety of clinical trials. In addition, we will describe the CMC information required in an IND. Lesson objectives. After this lesson, you will be able to describe the chemistry and manufacturing and controls (CMC) sections of investigational new drug application and clinical trial application. Learn the specifications and stability data required at the different stages of a clinical trial application. Describe the format of clinical trial application and investigational new drug application. Be able to discuss when process validation is required in a clinical trial application. Be able to describe the situations that lead to investigational new drug application and clinical trial application information amendments in CMC. Be able to describe the differences between the CMC section of investigational new drug application and clinical trial application. Clinical trial application. We will start with the CTA, which is required by Health Canada. The first part will deal with the chemistry and manufacturing of the drug substance. Introduction. Before sponsors can start a clinical trial in Canada, they must submit a clinical trial application to Health Canada and obtain approval according Division 5, C.05 of the Food and Drug Regulations and relevant guidance documents. Health Canada is receiving clinical trial applications in Non-Electronics Common Technical Document (ECTD) electronic-only format. The clinical trial application consists of Module 1, Administrative and Clinical Information. Module 2, Common Technical Document Summaries, Quality Information. Module 3, Quality, Additional Supporting Quality Information. We will only consider the chemistry and the manufacturing of the drug substance and drug product in this lesson. Let's start with the drug substance. Introduction. The sponsor should be able to provide most of the information on the drug substance, except the proprietary information found in the closed part of the drug master file. It is the responsibility of the sponsor to obtain all other information from the supplier of the drug substance and include this in the submission. The information from the open part of the DMF should be included in the quality overall summary. Part 1. Clinical Trial Application, Drug Substance. We will describe the information relating to the following in a clinical trial application: nomenclature, structure, general properties, manufacture, manufacturers, description of manufacturing process and process controls. Nomenclature. In this subsection, provide information on the nomenclature of the drug substance. For example, provide recommended international non-proprietary name (INN), compendial name, chemical name, company or laboratory code, other non-proprietary names such as national name, United States adopted name, USN, British approved name, BN, and chemical abstract service, CIS, or registry number. Structure. Provide the structural formula, including relative and absolute stereochemistry, the molecular formula, and the relative molecular mass when they are available or relevant. General properties. Provide a list of physicochemical and other relevant properties of the drug substance. The physical description should include appearance, color, and physical state. Manufacturers. In this subsection, provide the name, address, and responsibility of each manufacturer, including contractors, and each proposed production site or facility involved in manufacturing and testing of the drug substance. Description of manufacturing process and process controls. The manufacturing process description should be progressively more detailed from Phase 1 to Phase 3. Sponsors are expected to provide a flow diagram, accompanied by a narrative description for Phase 2 and Phase 3 only, summarizing the synthetic process of the drug substance. For drug substances which are manufactured as sterile substances, a complete description of the method of sterilization should be provided. As well, summarize controls in place to maintain sterility during transportation and storage. A flow diagram of the synthetic processes should be provided that includes chemical structures and configurations of starting materials, intermediates, and the drug substance. In addition, all reagents including chemical formulae, solvents and catalysts should be specified in the flow diagram. Part 2. Clinical Trial Application, Drug Substance. In the sections below I will describe more about the information to include in the clinical trial application. Control of materials. Controls of critical steps and intermediates. Elucidation of structure and other characteristics. Impurities. Control of materials. Drug substances are materials used in the synthesis which are of animal origin, should be free of bovine spongiform encephalopathy or transmissible spongiform encephalopathy. For sponsors of Phase 2 and Phase 3 clinical trial applications should provide details of the starting materials for the synthesis of the drug substance. The level of detail expected concerning controls on starting materials for synthesis required increase as synthetic steps get closer to the final drug substance. Controls of critical steps and intermediates. In this section no information is required for Phase 1 or Phase 2 clinical trial applications. For Phase 3 clinical trial applications, provide a summary of critical steps identified in the synthesis and the tests and tentative acceptance criteria for their control. You may also summarize in process controls or provisional specifications for isolated intermediates in this section. Elucidation of structure and other characteristics. For all clinical trial applications, confirmation of structure based on synthetic route and spectral analyses should be provided. Copies of the actual spectra are not required, but should be available if and when requested. The quality overall summary should include a list of the studies performed and a conclusion from the studies. Impurities. Impurities can affect the safety of a drug. Summarize the names, structures, and origin of the impurities in a table as described in a previous lesson. The origin refers to how the impurity was introduced, such as synthetic intermediate, or potential byproduct due to rearrangement during synthesis. Results of the impurity investigation should be provided with actual numerical results. Vague statements such as within limits or conforms should not be used. For Phase 2 and 3 clinical trial applications, the impurity name or identifier, structure if characterized and origin should be provided in the table for all specified impurities. Part 3. Clinical trial application, drug substance. I will describe the information required in the following sections of the clinical trial application. Specification. Analytical procedures. Validation of analytical procedures. Batch analyses. Justification of specification. For Phase 2 clinical trial applications, specifications are considered interim as they are based on a limited number of development batches. For Phase 3 clinical trial applications, specifications are expected to be reassessed prior to the Phase 3 application and reflect those intended for the marketing application, based on additional manufacturing experience and stability information. Analytical procedures. If your application is for Phase 1 clinical trial application, no information in this section is required. Provide a brief description of the analytical methods used for the drug substance for all tests included in the drug substance specifications. You don't need to provide detailed descriptions of the step-by-step analytical procedures for clinical trial applications. The descriptions should, however, be available upon request. Validation of analytical procedures. For Phase 2 and 3 clinical trial applications, the suitability of the analytical methods and a tabulated summary of the validation carried out should be provided. For example, provide results or values for specificity, linearity, range, accuracy, precision, intermediate precision, limit of detection and limit of quantification, where applicable. Complete validation reports are not required for clinical trial applications. Batch analyses. Provide a description of batches and results of batch analyses. The discussion of results should focus on observations noted for the various tests, rather than reporting comments such as all tests meet specifications. Batch analysis results for the drug substance may be provided in either the quality overall summary or by providing a copy of the certificate of analysis. The batch number, batch sizes, and dates and sites of production should be stated for all batches. Batch analyses. For Phase 1 clinical trial applications, Analytical results from the batches to be used should be provided. For Phase 2 clinical trial applications, provide analytical results from the batches to be used. If batch analysis from the actual batches to be used are not available at the time of filing, results from representative batches of drug substance may be provided as supporting data, with a commitment that the batch analysis for the specific lot will be submitted before dosing. Batch analyses. For Phase 3 clinical trial applications, provide analytical results from the batches to be used in the proposed clinical trial, or representative batches. Note: a representative batch is defined as a batch of drug substance that is manufactured using the same method of manufacture and equipment, specifications in the same container closure system as the proposed clinical batch, with a similar batch size. Justification of specification. If your application is for Phase 1 clinical trial application, no information in this section is required. Ensure the specification includes all the tests and acceptance criteria appropriate for the drug substance, and that reasonable limits for impurities and residual solvents have been established. Acceptance criteria should be based on manufacturing experience, stability data, and safety considerations. Part 4. Clinical Trial Application, Drug Substance. In this Part 4 of the presentation I will describe information for the following sections of the CMC section of the Clinical Trial Application: Container Closure System, Stability Summary and Conclusions, Stability Protocol and Stability Commitment and Stability Data. Container Closure System. Provide a description of the container closure system including the identity of materials of construction of each primary packaging component. Provide only a brief description for non-functional secondary packaging components such as those that do not provide additional protection. Provide additional information for functional secondary packaging components. Summarize the types of studies conducted, protocols used, as well as the results of the studies carried out. You don't have to provide full long-term stability data at the time of filing, provided you provide some preliminary stability data on representative batches together with a commitment that the stability of the clinical trial samples or representative batches will be monitored according to the stability protocol until you establish the retest period. Stability protocol and stability commitment. If you don't have full long-term stability data supporting the retest period at the time of filing, provide a commitment that the stability of the clinical trial samples, or batches considered representative, will be monitored according to the stability protocol. In addition, includes a summary of the stability protocol in tabular format, summarizing frequency of testing, tests to be conducted among others. Stability data. Provide brief description of the analytical procedure as well as a tabulated summary of validation information for Phase 2 and 3 clinical trial applications where analytical procedures are only used in stability studies and have not been described.
Let's now consider the drug product. Part 1. Clinical Trial Application, Drug Product. I will describe the information on the drug product to include in the clinical trial application under the following headings: Description and composition of the drug product, pharmaceutical development, manufacture, manufacturers, batch formula. Clinical trial sponsors should provide a description of the drug product and its composition. The information should include description of the dosage form, a list of all components of the dosage form, their amount on a per unit basis and a reference to their quality standards such as compendial monographs or manufacturer's specifications, description of reconstitution diluent if it's relevant. This section should contain information on the development studies conducted to establish that the dosage form, the formulation, manufacturing process, container closure system, microbiological attributes and usage instructions are appropriate for the purpose specified in the application. Pharmaceutical development. In situations where sterile, reconstituted products will be diluted further, compatibility must be demonstrated with all potential diluents covering the dilution range. These studies, required include tests for purity, potency, subvisible particulate matter, pH, and so on should if at all possible be conducted on aged samples. If the type of container for packaging is not specified, compatibility should be demonstrated in suitable containers. If one or more containers are identified, compatibility of admixtures should be demonstrated only in the specified containers. For Phase 1 clinical trial applications, sponsors should provide information for only sterile products. Include summaries of compatibility studies with diluents and containers in this section. Sponsors of Phase 2 and 3 clinical trial applications should to the extent possible provide information relating to pharmaceutical development such as discussion on the compatibility of the drug substance with excipients. If you have a combination product, provide a summary of investigations of the compatibility of the drug substances with each other. Provide a brief summary describing the development of the drug product taking into consideration the proposed route of administration and usage. Manufacturer. Provide the name, address, and responsibility of each manufacturer, including contractors, and each proposed production site or facility involved in manufacturing and testing. Batch formula. Provided a batch formula that includes a list of all components of the dosage form to be used in the manufacturing process, their amounts on a per batch basis, including overages, and a reference to their quality standards. You should declare the components by their proper or common names, quality standards such as USP, European Pharmacopoeia, in-house, and so on. You can also use their grades if applicable. I will describe under the following headings the information you need to include in the CMC section of a clinical trial application. Description of manufacturing process and process controls. Controls of critical steps and intermediates. Specifications. Excipients of human or animal origin. Novel excipients. The manufacturing process description should be progressively more detailed from Phase 1 to Phase 3. Provide a flow diagram, accompanied by a narrative description for Phase 2 and Phase 3 only and, summarizing the manufacturing process of the drug product. Provide a complete narrative description of the manufacturing process for sterile products, regardless of the phase of the trial. For Phase 1 clinical trial applications, a flowchart of the manufacturing process should be provided clearly indicating the order of addition of components and a summary of unit operations. In case of Phase 2 clinical trial applications, a flowchart and narrative description of the manufacturing process should be provided. The description of the manufacturing process at Phase 2 should be sufficient to fully describe the process without being restrictive to continuing process development and optimization. Controls of critical steps and intermediates. No information in this section is required for Phase 1 or Phase 2 clinical trial applications. Sponsors of Phase 3 clinical trial applications should to the extent possible provide information on the following. Test and provisional acceptance criteria for controls on the critical steps in the drug product manufacturing process. Information on the quality and provisional controls on intermediates isolated during the manufacturing process if relevant. This section includes the specifications for all excipients, including those that do not appear in the final drug product such as solvents. For excipients which are filed with Health Canada as a DMF, a letter of access should be provided as an attachment. Provide information for excipients of human or animal origin as well as information regarding adventitious agents such as sources, specifications, description of the testing performed and viral safety data. The information should include biological source, country of origin, manufacturer, and a brief description of the suitability of use based on the proposed controls. Note: for gelatin used in pharmaceuticals, supporting data should be provided which confirms that the gelatin is free of bovine spongiform encephalopathy or transmissible spongiform encephalopathy. If the supplier of the gelatin has a DMF registered with Health Canada, a letter of access should be provided. Novel excipients. In case of excipients used for the first time in a drug product or by a new route of administration, full details of manufacture, characterization and controls should be provided, with cross-references to supporting safety data using the relevant sections of the quality overall summary according to the drug substance and/or drug product format. The data may be non-clinical and/or clinical data. Part 3. The following important sections are also required in the CMC sections of the clinical trial application. Specifications. Analytical procedures. Validation of analytical procedures. Batch analyses. Characterization of impurities. You will not be required to submit information on specifications for Phase 1 clinical studies in this section. For other phases, provide a summary of the specifications for the drug product. For Phase 2 clinical trial applications, the specifications are considered interim as they are based on a limited number of development batches. In case of Phase 3 clinical trial applications, specifications are expected to be reassessed before Phase 3 submission and reflect those intended for the marketing application, based on additional manufacturing experience and stability information. Analytical procedures. You are expected to submit information for Phase 2 and 3, and not Phase 1 clinical trial applications. Provide a brief description of the analytical methods used for the drug product for all tests included in the drug substance specifications. You don't need to provide detailed descriptions of the step-by-step analytical procedures for clinical trial applications. The descriptions should, however, be available upon request. Validation of analytical procedures. Provide information for Phase 2 and 3, and not Phase 1 clinical trial applications. For Phase 2 and 3 clinical trial applications, the suitability of the analytical methods and a tabulated summary of the validation carried out should be provided. For example, provide results or values for specificity, linearity, range, accuracy, precision, intermediate precision, limit of detection and limit of quantitation, where applicable. Complete validation reports are not required. For substances which comply with the Schedule B monograph, reference to the monograph is considered adequate. Provide a description of batches and results of batch analyses. The discussion of results should focus on observations noted for the various tests, rather than reporting comments such as all tests meet specifications. Batch analyses. For Phase 1 and Phase 2 clinical trial applications, analytical results from the batches to be used in the proposed clinical trial should be provided. For Phase 2 clinical trial applications, if batch analysis from the actual batches to be used in the proposed study are not available at the time of filing, results from representative batches of drug product may be provided as supporting data. However, a commitment that the batch analysis for the specific lot to be used must be submitted before dosing. In case of Phase 3 clinical trial applications, you should provide analytical results from the batches to be used, or batches considered representative. Provide information on the characterization of impurities that has not been submitted previously in a summarized format. This section may also be used to report any new impurities found in the drug product during stress testing, such as during phytostability testing. In this last section, I will describe the type of information to include in the CMC section of the clinical trial application under the following headings. Justification of specifications. Container closure system. Stability. Stability summary and conclusions. Stability protocol and stability commitment. Stability data. Justification of specifications. Submit information for Phase 2 and 3, and not Phase 1 clinical trial applications. You should ensure that the specifications include all the test and acceptance criteria appropriate for the drug product, and that reasonable limits for degradation products have been established. Acceptance criteria should be based on manufacturing experience, stability data, and safety considerations. For impurities or degradation products which are unique to the drug product, acceptance criteria should be supported by appropriate container closure system. Provide a description of the container closure system, including the materials of construction for each packaging component. In case of sterile products, submit details of the washing, sterilization and removal of pyrogens. More details may be required for dosage forms such as parenteral, ophthalmic products and oral solutions which have a higher potential for interacting with filling and container closure system. Stability summary and conclusions. Provide a summary of the types of studies conducted, protocols used, and the results of the studies. You are not required to submit full long-term stability data at the time of filing, provided you present some preliminary stability on representative batches. However, you need to provide a commitment that the stability of the clinical trial samples or representative batches will be monitored according to the stability protocol until the shelf life of the drug product has been established with confidence. Stability protocol and stability commitment. If you don't have the full long-term stability data to support the proposed shelf life at the time of filing, you can provide a commitment that the stability of the clinical trial samples, or samples considered representative of the clinical batches, will be monitored throughout the duration of the clinical trial. You will need to submit a summary of the stability protocol in tabular format, summarizing frequency of testing, tests to be conducted, and so on. Stability data. Present the results of the stability studies such as long-term and accelerated studies in an appropriate format. The actual stability raw data results should be provided as an attachment. For quantitative tests, such as an individual and total degradation product tests and potency tests, ensure that actual numerical results are provided and not statements such as within limits or conforms. In case of Phase 2 and 3 clinical trial applications, where analytical procedures are only used in stability studies and were not previously summarized, details of the analytical procedure as well as a tabulated summary of validation information should be provided.
In this section I will describe briefly the type of changes in the CMC section of a clinical trial application that must be submitted to Health Canada for review. Please note that these amendments apply to only approved clinical trial applications. They don't apply to unapproved applications. Sponsors must file a clinical trial application amendment for chemistry and manufacturing changes that may affect the quality or safety of the clinical trial drug supplies. Changes include the following. Pharmaceutical drugs. Manufacture of the drug substance, where new ingredients are used, including ingredients which do not appear in the final drug substance, and where a new impurity or degradation product has been identified.
Clinical trial application amendments continued.
Control of drug substance, where a test is removed from the specification, and/or the test methods or limits are relaxed. Manufacture of the drug product, where new ingredients are used, including ingredients which do not appear in the final product. Description of manufacturing process and process controls, for sterile products only where the sterilization process is changed. Control of drug product, where a test is removed from the specification, and/or the test methods or limits are relaxed.
Clinical trial application amendments continued.
For biologics and radiopharmaceuticals. Manufacture, regarding raw materials, where a new medicinal or non-medicinal ingredient is used, including ingredients not appearing in the final formulation.
Clinical trial application amendments continued.
In control of drug substance, where test method is altered and/or the test limits are relaxed. In manufacturing, where minor changes to the formulation process are proposed. Where a test method is changed and/or test limits are relaxed. In the control of drug product, where an extension of the shelf life for drug product is proposed, and the original expiry date granted was less than 18 months, and where modifications to an existing facility are proposed.
Now we will describe the information to include in the CMC section of the investigational new drug application. We will describe required information for the drug substance and drug product. Please note the similarities of the content in the clinical trial application. Introduction. Sponsors are required to submit investigational new drug application before they start a clinical trial in USA. In this part of the lesson you will learn the type of information in an investigational new drug application required at different phases of the clinical trials. How the amount and depth of CMC information that would be submitted to the FDA depends on the phase of the investigation, the testing proposed in humans, and its relationship to safety. Situations that might trigger information amendments in CMC. At the beginning of the CMC section, the sponsor should state whether it believes the chemistry of either the drug substance or the drug product, or the manufacturing of either the drug substance or the drug product, presents any signals of potential human risk. Required information depends on the clinical trial phase. The amount of information to be submitted depends upon the scope of the proposed clinical investigation. The amount of information submitted also depends on the phase of the clinical trial. As drug development proceeds and as the scale or production is changed from the pilot scale production to the larger scale, the sponsor should submit information amendments to supplement the initial information submitted on the chemistry, manufacturing, and control processes with information appropriate to the expanded scope of the investigation. I will tell you more about information amendments later. Dealing with potential risks of experimental drugs. If there are signals of potential risks due to the drug substance or product, these signals should be discussed. Steps should be proposed to monitor for such risk. Steps should be described. Alternatively, the reasons for why the signal are dismissed should be discussed. Differences in drug product in animal studies with drug product in human studies. What if there are differences between the drug product proposed for clinical use and the drug product used in the animal toxicology trials? Sponsors should describe any chemistry and manufacturing differences between the drug product proposed for clinical use and the drug product used in the animal toxicology trials that formed the basis for the sponsor's conclusion that it was safe to proceed with the proposed clinical study. Justify differences in drug products. Differences between the drug product proposed for clinical use and the drug product used in the animal toxicology trials may or may not affect the safety of the drug. How these differences might affect the safety profile of the drug product should be discussed. If there are no differences in the products, it should be clearly stated. Drug substance. Sponsors may use the USPNF to satisfy some of the requirements of the drug substance. Information on the drug substance should be submitted in a summary report as described in the slides that follow. Description of the drug substance. Provide a description of the drug substance, including its physical, chemical, or biological characteristics. A brief description of the drug substance and some evidence to support its proposed chemical structure should be submitted. The FDA acknowledges that the amount of information about the structure will be limited in the early stage of drug development. The amount of information will vary with the stage of development. Manufacturer information. The name and address of its manufacturer. The full street address of the manufacturer of the clinical trial drug substance should also be submitted. Preparation of the drug substance. Submit a brief description of the manufacturing process, including a list of the reagents, solvents, and catalysts used. Use a detailed flow diagram to present the information as it is the most effective means of presentation. Where the drug is derived from biotechnology or extracted from human or animal sources, more information may be needed to assess its safety. Use the acceptable limits and analytical methods used to assure the identity, strength, quality, and purity of the drug substance. Description of the test methods. Submit a brief description of the test methods used. Provide proposed acceptable limits supported by simple analytical data, such as infrared spectrum to prove the identity, and HPLC chromatograms to support the purity level and impurities profile of the clinical trials material. You can also submit a copy of the certificate of analysis. Note that the specific methods will depend on the source and type of drug substance, because products of animal source, plant extract, radiopharmaceutic, other biotechnology-derived products will require different scientific approach. Validation data and established specifications. You will normally not need to submit validation data and established specifications at the initial stage of drug development. You may, however, need to submit preliminary specifications and additional validation data in certain circumstances to ensure safety in Phase 1 clinical studies for some well-characterized therapeutic biotechnology-derived products. Stability data for proposed clinical studies. Information to support the stability of the drug substance during the toxicologic animal studies and the proposed clinical studies is required. Submit a brief description of the stability study and the test methods used to monitor the stability of the drug substance. Submit preliminary tabular data based on representative material. Neither detailed stability date nor the stability protocol should be submitted.
Drug product. Sponsors are reminded that, under present regulations, references to the current edition of the United States Pharmacopoeia National Formulary may be used to satisfy some of the requirements, when applicable. Information on the drug product should be submitted in a summary report containing the items in the following slides.
Listed components. A list of all components may include reasonable alternatives for inactive compounds used in the manufacture of the investigational drug product, including both those components intended to appear in the drug product and those which may not appear, but which are used in the manufacturing process. List of drug product components to be provided. Submit a list of components of up to 2 pages of written information. The quality of the inactive ingredients, given as NF, USP, ACS, should be cited. If novel excipients were used, additional manufacturing information may be required. Composition of the investigational new drug product. Where applicable, the quantitative composition of the investigational new drug product, including any reasonable variations that may be expected during the investigational stage, is required. Submit a brief summary of the composition of the investigational new drug product. Information on component ranges is not necessary in most cases. Submit the full street address of the manufacturer of the clinical trial drug product. Method of manufacturing drug product. A brief, general description of the method of manufacturing and packaging procedures as appropriate for the product is required. Submit a diagrammatic presentation and a brief written description of the manufacturing process, including sterilization process for sterile products. Analytical methods and acceptable limits. The acceptable limits and analytical methods used to assure the identity, strength, quality, and purity of the drug product are needed. Analytical methods and acceptable limits. Submit a brief description of the proposed acceptable limits and the test methods used. Tests that should be submitted will vary according to the dosage form. For example, for sterile products, sterility and non-pyrogenicity tests should be submitted. Submission of a copy of the certificate of analysis of the clinical batch is also suggested. At the initial stage of drug development, validation data and established specifications need not be submitted. Adequate assessment of bioactivity and preliminary specifications should be available in the case of our well-characterized, therapeutic, biotechnology-derived products. Stability study and the test methods. Submit a brief description of the stability study and the test methods used to monitor the stability of the drug product packaged in the proposed container and container closure system, as well as storage conditions. You may submit preliminary tabular data based on representative material. Detailed stability data or the stability protocol are not required. Composition, manufacture and control of placebo. Provide a brief general description of the composition, manufacture, and control of any placebo to be used in the proposed clinical trials. Submit a diagrammatic, tabular, and brief written information for manufacturing the placebo. Copy of all labels and labeling. Submit a copy of all labels and labeling to be provided to each investigator. Submit a mock-up or printed representation of the proposed labeling that will be provided to investigators in the proposed clinical trial. All investigational labels must carry a caution statement that reads, "Caution, new drug, limited by federal or United States law to investigational use." Please ensure that there are no false or misleading statements and no representation that drug is safe or effective for indicated use. Environmental assessment and exclusivity requirement. Submit a claim for categorical exclusion from or submission of an environmental assessment. FDA acknowledges that most products may qualify for a categorical exclusion. Sponsors who believe their investigational product qualifies for exclusivity should submit a statement certifying that their product meets the exclusivity requirements and request a categorical exclusion on that basis. Option 1. This slide shows that your CMC section of IND consists of Module 2 and 3. You can provide either a full quality overall summary or one document under 2.3 Introduction. Module 3 will consist of documents under drug substance and drug product as well as appendices and regional documents. You may provide one or more documents for each CTD heading. Option 2. This slide shows that your CMC section of IND consists of only Module 2. This module will consist of documents under Introduction, drug substance and drug product as well as appendices and regional documents. You can provide single or multiple documents under each heading depending on the choice of attributes. This might be suitable for early stages of drug development. Option 3. In this option there is no content in Module 3. All the CMC information is captured in 2.3 Introduction, 2.3 Drug Substance, 2.3 S.1 to 2.3 S.7 with 7 documents, 2.3 Drug Product, 2.3 P.1 to 2.3 P.8 with 8 documents, and 2.3 appendices. These sections are only summaries during the early phases of development and thus kept in Module 2. Option 4. This option is a modification of Option 3 with only batch analysis and stability data sections of data added in Module 3. CMC information is captured in Module 2. Option 5. In this option you provide only the introduction in Module 2. Module 3 consists of the drug substance, drug product and appendices. You may provide a summary under each heading in Module 3.
Information amendments. Once the IND is approved, you can submit information amendments because of certain situations not anticipated during the application of IND. Information amendment. An information amendment should be submitted that describes the changes and contains relevant information at a level of detail sufficient for an adequate review and assessment of the following changes: the synthetic pathway used to manufacture the drug substance, material change in one of the bond-forming steps, change in a solvent used for the last reaction, or crystallization step, change resulting in a different impurity profile. Information amendment, manufacturing process. Change in the manufacturing process that can affect the quality of a drug substance produced by fermentation or derived from a natural source such as plant, animal, or human. Change in the manufacturing process that can directly or indirectly affect viral or impurity clearance for a drug substance produced by fermentation or derived from a natural source. Change in the manufacturing method from one manufacturing method such as chemical synthesis, fermentation, or derivation from a natural source to another. Information amendment, source of raw material. Change of source material such from as plant to animal, species, part used or country of origin for a drug substance derived from a natural source. Change of species and or strain of microorganism for a drug substance produced by fermentation. Information amendments, specifications. The following changes to specifications may result in information amendments. Change of the method of sterilization of the drug substance or drug product. Change of the route of administration. Change of the composition or dosage form of the drug product. Change the drug product manufacturing process that can affect product quality. Change in the drug product container closure system that can affect product quality such as metering capability or dose delivery.
We have come to the end of the presentation by Royal Canadian College of Distance Education. Thank you for listening.
See you in the next class.